the procedure for dealing with a microbial or chemical spill. So the very first thing, if you have a spill of microbial broth culture or chemical, just don't be embarrassed. Shout out, we have a spill of, and then let us know what's been spilled. Um, I will call back, is everybody okay? And so, of course, we're hoping that everybody's safe, nobody's been hurt, there's been no splash to the eyes. So if everybody is safe, we would like the people involved in the spill to stay put. And that's to prevent you from tracking the chemical or the microbial culture around the lab. So if everybody's safe, we would like you to stay put, and then we will come and help you. However, if there's been exposure to your eyes, we want to take you immediately to the eye wash station in the back of the lab, or if there's full body exposure, we'll take you into the autoclave room where there's a full body shower. If health is in danger or life-threatening, we would call the campus police and 911. If, however, no one is hurt, which is, of course, what we're hoping for, again, we'd like you to stay in place. We will come and help you. And what we'll do is we're going to cover the entire spill area with paper towels and spray the spill area with disinfectant. The paper towels prevents the liquid from spreading further, and it also is a visual indicator where the spill zone is so other people won't walk through it. We try to put the paper towels down first before spraying with disinfectant so we won't get any splashback from the disinfectant. Kill time, so the concept of kill time, the longer the disinfectant is in contact with the microbes, the greater will be the percentage kill of the population of microbes. So longer is better. We want to leave the disinfectant in contact with a microbial spill for 15 minutes at least, if not longer. And then usually I try to do this. I try to be the one responsible for the cleanup. Then in full PPE, we would gather all of the paper towels and place them in the biohazard bag. The reason for this is anything that's come in contact with a biological hazard, we never know if the disinfectant has totally killed all the microbes. So all potentially contaminated paper towels would go into the biohazard bag at the end of the bench. If there was any glass involved, again, your instructor would clean that up with a special dustpan and broom. And glass is going to be placed in the sharps container in the kill area. After cleaning up the paper towels and any glass, we're going to spray the spill area again with disinfectant. Following that, the person involved in that cleanup would remove their gloves and place them in the biohazard bag. We would wash our hands, always, the very last thing we do is always to wash our hands. In addition, during the first week of lab, we will show you the location of the chemical spill kits. And again, we would like the instructors to deal with microbial or chemical spills. We're lucky in that our campus budget committee gave us money to buy Incinerators. This has greatly reduced the incident of burns or little mini fires on the bench tops that were a result of accidents when we were using our open flame Bunsen burners. However, the Incinerators, there are some safety issues involved, so please watch the short video here that we made. A couple of things to remember is the Incinerators, once they're turned on, the barrels will be incredibly hot. So make sure you never touch the Incinerator when it's turned on. The Incinerator will remain really hot even after you turn it off. So make sure, just don't touch the Incinerator barrels ever. When you sterilize your loops or needle using the Incinerator, the loop or needle will be incredibly hot. So never ever touch the wire portion of the loop or needle, and also never put the loop or needle down on the bench, especially after you've just sterilized it, because they'll be so hot they will ignite paper. We had a benchtop fire last semester from a hot loop being placed on a piece of paper towel. So do watch this Incinerator video. I think there is one mistake in the video, and I think I misspoke. I think I said the needle might be used to stab an auger deep so the bacteria would grow aerobically, and that's incorrect. when we use our needles to stab auger deeps to get our microbes growing in the butt, that's to force them to grow anaerobically without oxygen. So we'll try to repeat that in lab. Fire is a big, big safety issue. So if we are ever using our Bunsen burners, never walk away from them when they're lit. Likewise with a hot back tea incinerator, please try not to walk away from a hot back tea incinerator, even for a few seconds. If you must, if you have to walk away from your back tea incinerator, please put a little sign in front of it saying "Caution Hot," so another person doesn't accidentally come along and touch it. Really important to keep your benches really clear. Clear them of all flammable objects before using your back tea incinerator or Bunsen burner. Tie back your long hair. Again, step 4 here, into the hot back tea incinerator, only insert the wire portion of your looper needle, and we'll demonstrate this. You're going to move the looper needle by handling the end of the handle. Never put your fingers near the tip of the looper needle because you will burn yourself. Never place a hot looper needle on paper on the bench top. As we said, the paper will ignite, will have a fire. And again, just remember that back tea incinerator barrel will remain hot even after you turn it off. So please don't touch the barrel. In lab, we'll go over location of our fire blankets, the fire extinguisher, the fire water buckets, and the shower. The location of the fire alarm, the fire alarm is located on the right-hand wall as you enter Lillard Hall through the double glass doors. And we will be talking about our evacuation route and where we would meet for roll call if we ever have an evacuation. We'll probably take a little walking trip, so everybody's familiar with that. If there is a fire and no humans are harmed, obviously if it's a large fire, we're going to evacuate our lab, meet in our meeting place. We'll hit the fire alarm at the entrance of Lillard Hall. Our evacuation meeting is the southeast corner of the LRC, the library. We'll be walking there, and it is important that we meet there so we can do roll call to make sure everybody got out of the building. In the past, when we have had little mini fires on the benches or in the sinks, again, you want to shout out fire and indicate where the fire is. We want to turn off our Bunsen burners, turn off our back tea incinerators. If it's a little tiny fire, then we'll try to extinguish it. We have multiple ways to do that. We have fire blankets we'll show you. We have a fire extinguisher near the doorway into the lab. We'll have small and large fire buckets located around the lab. Some of the small fire buckets we'll put in the sinks. We want to make sure that the fire buckets are filled with water whenever we're working with Bunsen burners or with our back tea incinerators. And importantly, folks, don't handle burning objects with your bare hands. That's happened in the past, and it's not good. You're going to get burnt. So if it's a significant fire and somebody's been burnt or somebody's clothes catch on fire, again, the first thing is just to call out fire, X is on fire, my colleague's on fire. So to help extinguish the flames, often the person whose clothes are on fire will be panicked and they'll be running, and that's providing more oxygen to the flames. So we want to do the stop, drop, and roll. We want to help to get them down on the ground, have them cover their face, and then we want to roll them, trying to extinguish the flames. And again, we want to use the fire blanket. We could use water from our water buckets. In an emergency, you can take off your lab coat and use a lab coat to try to extinguish the flames. As soon as the flames are out, we take our colleague into the full body shower in the autoclave room and start the cooling process of showering. And of course, we've called campus police and 911. A new topic, but again, very important safety topic, is the topic of microbial cultures and labels. So it's so important when we're growing microbes in the lab that we know which microbe is growing where. So labels are so very important. If we are labeling agar plates, growing our microbes in agar plates, we can write directly on the bottom of the plate, preferably with a thin tip permanent marker. If we are growing our microbes in glass test tubes, We don't write directly on the glass test tubes as they get reused over and over, so we use masking tape. Now, it's really important to know the important information that goes on a label, and this is usually on our lab quiz or on our lab exams. So the first thing on your label, you want to write the name of the microbe. Then you want to write your first initial and your full last name. We always have to track back who's the microbiologist responsible for that culture. You put the date and you put the lab section. For example, I would put Monday, Wednesday, 8 a.m. is my lab section. Secondly, it's really important to know where the labels should go. So when we're growing microbes in an agar plate, we want to place the label on the bottom of the plate. And the bottom of the petri dish contains the agar, and we're going to place our microbes on top of the agar. So we always want to place the label on the bottom of the agar plate, the petri dish, because that's the part of the petri dish the microbes are growing in. On test tubes in which we're growing our microbes, we don't put the labels on the caps because caps can fall off, they can get mixed up. We always put the labels on the glass bodies, as we see here, right here. Okay, so just remember the label always goes on the part of the container in which the microbes are living and growing. We will demonstrate in lab that once we've inoculated our agar plates, we will use masking tape to tape the lid to the bottom. We'll make two little masking tape hinges. And then when we're going to incubate the plates, keep them warm by placing them in our incubators, we're going to invert them, which means we're going to turn them upside down. So we can see here, so the label would be pointing upward, and that's really helpful when we have hundreds of agar plates in the incubator. It's really helpful that you can see right off the bat which microbe is growing in which agar plate. When we're finished using the agar plates, you don't have to take any tape off. The agar plates will be disposed of in the biohazard bins, the autoclave bins. They'll be autoclaved and then thrown directly into landfill, so we don't have to take the tape off them. However, as we mentioned, the test tubes in which we grow our microbes, they will be autoclaved and then washed and reused. So for our microbial cultures grown in test tubes, we have to remove all of the tape. Very importantly, in our lab, most of our test tubes for our microbial cultures are what we call screw cap tubes. So it is so important that you loosen those screw caps before you put them in the incubator, before you put them in your locker, before you put them in the kill area. So we want to discuss that in greater detail. So this is an example of the type of tubes that we'll grow our microbes in. So this is a screw cap tube we see here. And the process that we need to be aware of is when we inoculate our microbes into the media in these tubes, whether it's broth or an agar slant or an agar deep, the microbes, as they start growing and dividing, they often produce huge amounts of gas. And this creates a real safety issue. If you tighten these caps and the microbes are making large volumes of gas, then this test tube becomes really a little micro hand grenade. The gas pressure can build up to such a degree that when you go to move the tube, you might bump it. It could explode. And in fact, this has happened in our lab before, where a person forgot to loosen the screw cap of a tube that was put in the kill area. And then when the tube was moved, it exploded in the person's hand and slit their hand open and required several sutures there. So always double check to make sure that you've barely loosened the screw caps so those gases can vent so they can escape. So what I usually do is I'll tighten the screw cap and then just barely loosen it. We don't want the caps falling off. That's a safety hazard as well. But you just want to have a little bit of movement on that cap so you know that the gases can vent. Historically, people always knew that if you had canned food and the lid of the can was bulging outward, you didn't want to eat that food because that bulge indicated that microbes were growing in the container, forming large volumes of gas. And the big one, of course, was always Clostridium botulinum, causing botulism. For our different lab experiments, we'll have different waste products, and there'll be a specific place for all of those waste products. In general, anything that could have microbial contamination has to be autoclaved. So it's either going to be going into our autoclave bins, autoclave bags, Um, uh, Tubes with microbial cultures will go into the kill area. There's specific bins for auger slants and auger deeps, and a separate bin for auger broths. Any paper that's potentially been contaminated with microbes also goes into autoclave bags. Any glass goes into the sharps container in the kill area. So again, with each experiment, we'll try to make sure we go over proper disposal. And if you have any doubts, please ask us. Ask your instructor. The most important thing to keep us all safe in lab is just ask questions. Never feel embarrassed. Um, Never hesitate to ask a question. And very often the question you have is the question all the rest of your colleagues have. So please never hesitate to ask a question. And just remember, the most important thing in the micro lab is to keep ourselves and our classmates, our colleagues safe. This last slide, these are a couple of very outdated videos. So maybe only if you don't have anything else to do, you could take a look at these videos, but a lot of it is outdated, and some of the information we no longer do in our Sac City Micro Lab. Okay, so we'll be moving on to Lab Chapter 2, Unit 2, and that's materials and equipment that we use in the micro lab.